All right, now let's talk about amino acids, how they are made, mostly in humans. And what we need to start with is that they are actually muscle is an excellent source of food, and they also constitute proteins amount to about half of the cell's mass. The amount of protein you have in every cell defines almost the growth capacity of that cell. So, and the useful metric of cell growth is the amount of protein that's in that cell per generation. How do they make these amino acids? How do the cells make these amino acids? If we don't get them into our diet, depending on the organism, some are essential, some are non-essential. The essential amino acids have to be provided into the diet. But all organisms can make at least some amino acids, maybe not all, but some. The source, the building blocks for making these amino acids, we keep talking about them through the course, throughout the course. When we're discussing glycolysis, we said they are building blocks for for amino acids. So pyruvate is the source for valine, valine, isoleucine, and leucine. 3-phosphoglycerate makes serine glycine, and from there you can make cysteine. The pentose phosphate pathway makes ribose 5-phosphate. Okay, that's a very important pentose that is made through the pentose phosphate pathway, and that can be used to make nucleic acids, but also one very important amino acid, histidine, and as you will see, provides another intermediate to make aromatic amino acids. The TCA cycle, many amino acids are use TCA cycle intermediates to make as a building block during their synthesis, and two very important ones are alpha-ketoglutarate, the 5-carbon compound that gives glutamate, glutamine, proline, lysine, and arginine, and the 4-carbon group that comes from oxaloacetate gives this set of amino acids. The aromatic amino acids, phenylalanine, tyrosine, and tryptophan are special. They, as you'll see, they have a very complex and long series of steps during their biosynthesis, and they use both pentose phosphate pathway and glycolytic intermediates. So they use phosphoenol pyruvate, a very super high energy compound from glycolysis, and they use also erythrose 4-phosphate, which we saw in the pentose phosphate pathway, in these transketolase reactions. Everything in red we cannot make. So we humans cannot make everything that's shown is in red here. So those are essential. They are almost like vitamins, right? Another word for it. So we have to eat them through our diet. The black ones we can make. If you look at the reactions of amino acid biosynthesis, and I just preempt you here, they can get very complicated. Okay, so I try to do my best to categorize them and give them in groups, but there is no denying that amino acid biosynthesis is complex. There's going to be a lot of reactions, a lot of arrows. It's not like the previous catabolic reactions we had seen in in glycolysis or TCA cycle that kind of makes sense. Okay, it's a limited set of reactions. Here we have we're dealing with a lot of variety, lots of flavors because there are many of them, right? If you take a survey of how many ways are there to make amino acids, there are quite a few depending on the organism. And about 3.5 billion years ago, let's say 2 billion years ago, the reactions were already there. They were set. Okay? And they come in different flavors depending on the organism for most of them. Some of them have lost their ability to make amino acids, like our precursor, about 2 billion years ago, some eukaryote lost some of this capacity. Depending on the organism you are. Now, with the exception, so this variety in ways of making amino acids, we're not going to cover all of them. Okay, we're only going to focus on the human or the ones that are important for human biology. But there are many ways for most of them, except the aromatic amino acids. To make aromatic amino acids, the organisms that can make them, they always make them in the same way. So aromatic amino acid synthesis is very complex. As I said, one good solution in these eons came along, and that was the best one, and it's got fixed, and it hasn't been improved since. Okay. Before we describe these reactions in more detail, it's useful to explain how people figured out how amino acids are put together, okay, the biosynthetic mechanisms. And this goes way back and has to do with tracer experiments. So the idea here is you give the cells a precursor, very simple, like CO2 here, which is labeled, it's radioactive, let's say, or heavy, some isotope of some sort, right? And then you follow where that ends up. And then you can, with some clever deduction, and it requires extreme knowledge of all the chemical reactions in the cell of metabolomes, you can figure out and guess the pathway that leads to the formation of an amino acid, depending on how much of this tracer, of this radioactivity, let's say, per mass is found on the product. So you start with the reactant, very simple, and you follow where that black label goes into the product that you care about. So if you start, let's say, with CO2 and pyruvate, so if you carboxylate pyruvate, how would your product look like? Well, the product would be oxaloacetate, and this extra carbon that is labeled, you give it you gave it a labeled carbon, let's say C14 in this case, radioactive. It will be on oxaloacetate, okay? And we can depict this in the circles like this, okay? So very simple. Now, the key to remember here is all the reactions, however, right? So is that what you're going to see when it goes, you make oxaloacetate, but what happens to oxaloacetate? Oxaloacetate will be going through the TCA cycle. So when you go find oxaloacetate and you take it out of the cell, so you can look where the carbon, the labeled carbon went, okay, what you're going to find is something different. It's going to find that it's, now the carbon is, the label is all there on oxaloacetate. So the amount of radioactivity on one carbon, radioactive carbon, is found, all of it, on oxaloacetate, but it's spread onto two different carbons. And you start scratching your head and say, okay, why? Why is that happening? I mean, you carboxylate, you should be getting this. Why are you getting 50-50? Okay. So instead of 100% of the carbon radioactivity being on one carbon on oxaloacetate, you find that it's actually spread equally on two carbons. And if you go back to your reactions, you know that once you make oxaloacetate, so pyruvate comes in, okay, 3 carbon with CO2, you make this oxaloacetate here, but doesn't stay like that. It goes around through one cycle, okay, and eventually it's going to be scrambled. So that's why it's like this. So but you do make oxaloacetate, okay, in this form, it's labeled. And if you give it some acetate, so 2 more carbons, you find that you will make aspartate methionine and also isoleucine. The last step is with the 2 extra carbons going to be on isoleucine. From oxaloacetate, you can make straight these compounds. And those, all of them, will have the same specific activities. So what does that mean? Specific activity is the amount of radioactivity per mass of the molecule you follow. If that ratio is the same, okay, it stays, all of the radioactivity stays in the molecule, then it's going to have the same specific activity as the C14 carbon dioxide that you gave in the beginning. So oxaloacetate seems to be straightforwardly converted to all these amino acids eventually. If you go through the TCA cycle again and you say, okay, this is what I have with oxaloacetate, and then you keep going with you give it acetyl-CoA, you have the acetate, which is unlabeled. As we've seen, you're going to lose 1 carbon in order to get to alpha-ketoglutarate. So oxaloacetate plus this 2 will give you citrate, and then goes through. and that one carbon, one of those guys will be gone. So what you're going to end up having is half of the radioactivity that was on the CO2 will be found on alpha-ketoglutarate. Okay, so you have a drop of 50% the specific activity, all right, because you lost this carbon there. And then you follow what other amino acid has the same specific activity as alpha-ketoglutarate. Okay. So let's recap. CO2, let's say, has 100 specific activity. Oxaloacetate would also have 100, split equally on 2 carbons. And the same 100 specific activity will be found on these amino acids, aspartate, methionine, threonine, and isoleucine, suggesting that oxaloacetate was the source of these amino acids. Now, alpha-ketoglutarate is not an amino acid, okay, it's a TCA cycle intermediate, but because you lose 1 carbon, it's going to be 50%. It's going to be a specific activity of 50 if CO2 was 100. And anything else that has a 50, such as glutamate and proline, you can presume that it came from alpha-ketoglutarate, all right? And you keep going, and that's really the idea. Now, sometimes you find more, okay? So you have 150 here for arginine because there is an extra carboxylation. Okay? So that you can devise in your scheme and say, okay, that can also should also come from alpha-ketoglutarate. You combine the 50 plus the 100, that's straight with 1 carboxylation, and you get arginine. So that's also alpha-ketoglutarate is also a precursor for arginine, not just glutamate and proline, right? Vice versa, if you lose this singly labeled carbon on alpha-ketoglutarate, you get 0. Okay, so lysine will have a 0 because there is a decarboxylation. And it is through this scheme, this approach that most reactions have been figured out of amino acid biosynthesis. So before we describe them in detail, I thought it was interesting to tell you how people came up with these schemes and these reactions of how amino acids are made. Originally, they traced the fate of carbons from CO2, let's say in this case, all the way to specific amino acids. And you see how that fate changes the specific activity depending on what intermediate was used, let's say oxaloacetate or alpha-ketoglutarate, to make this specific amino acids. We will stop at this point, and we will pick up with the synthesis of amino acids in the next video. And we're going to discuss the alpha-ketoglutarate group, which, as I said, covers glutamate, glutamine, proline, arginine, and lysine, which you see here. Okay, we haven't talked about glutamine, but you'll see how that also comes straight from glutamate. All right, so I will see you next time in the next video.